Aposec path to clinical application. Aposec is the secretome of stressed white blood cells turned PBMCs containing everything these blood cells release to the culture medium. In the event of illness or injury, these substances help to recover and regenerate damaged tissues. Aposec is a biological drug that helps repairing damages where the blood supply is not working properly, inducing hypoxic damages. Such damage can be found, for example, after stroke or heart attack, spinal cord injury, and non-healing wounds. White blood cells are an integral part of our immune system and currently a waste product of blood donation. Therefore, Aposec is manufactured in a blood donation center. The Red Cross Blood Transfusion Service for Upper Austria and Linz produces Aposec for the company Aposcience under the strictly regulated conditions of Good Manufacturing Practice (GMP). Aposec is then released by a qualified person. Aposcience is a clinical stage company working according to the guidelines of the International Council for Harmonization. Aposec is an investigational medicinal product currently being tested in a clinical trial. The road to market approval for a new drug is a long one. PBMCs are separated from blood, irradiated in culture medium, and incubated for 24 hours. The supernatant containing the secretome is separated from cells and debris, is filtered and subjected to virus clearance. The culture medium containing the secretome is lyophilized and terminally sterilized by high-dose irradiation. The cell-free lyophilized supernatant represents the final investigational medicinal product. Aposec was investigated in preclinical research studies on 3 different levels. First, Aposec was tested in animal models whether the investigational medicinal product works. Then, in vitro studies demonstrated the mode of action. And finally, product characterization was done according to good manufacturing practice. This was done in a laboratory using molecular and cell biological methods. These 3 pillars are the roadmap for drug development. After that, the safety of the new drug must be examined in small and large animal models under the strictly controlled conditions of good laboratory practice (GLP). These pharmacological and toxicological studies are prescribed by the authorities and primarily serve the safety of the patient. Toxicological programs for Aposec were designed for topical and intravenous applications. Therefore, the safety for human application was demonstrated in 4 species, namely mice, rats, minipigs, and macaques. Methods for viral clearance for Aposec were validated under good laboratory practice conditions to establish methods ensuring product safety. In addition, stability programs for Aposec were performed and long-lasting shelf life was proven. Last, validated biological assays, so-called potency assays, were developed to prove the mode of action of Aposec and batch-to-batch comparability. When the production of Aposec according to good manufacturing practice meets the product specifications, the qualified person of Aposcience can release the investigational medicinal product. Only when these studies have been successfully completed, health authorities may approve the clinical trial in a certain indication, such as diabetic foot ulcer or stroke, with a certain drug formulation. Then, the drug can be tested on humans for the first time after passing clinical trial evaluation by the national and supranational authorities. The first clinical application for Aposec is the topical treatment of diabetic foot ulcers, MASIAS 2, and received approval from the health authorities and ethics committees of Austria, the Czech Republic, Germany, and Poland. A phase 1 trial was concluded in 2021 and the Data Safety Monitoring Board has unequivocally suggested continuing with the phase 2 trial. 120 patients with diabetic foot ulcers will be enrolled. The trial meets the high scientific standards of good clinical practice (GCP) and is monitored by a contract research organization. MASIAS 2 is a randomized, double-blind, placebo-controlled phase 2 trial. We are currently planning a phase 1 clinical trial for intravenous application of Aposec. It is the central aim of ApoScience to harness the secretome of stressed white blood cells, a current waste product of all transfusion units worldwide, in order to provide therapeutic benefits to patients with unmet needs.